and I was the first year student. I used to see one student used to get all medals, physiology, gynecology every year. And he published an article when he was doing postgraduate in International Ophthalmology, International Journal of Ophthalmology, when he was a postgraduate in 1980s, 80s, no, before that, 90. So he was a pole star to us, many of us, he was a pole star. We try to combine the basic and clinical research, seeing him. And when I went to USA, the first person who called me was Arup, and he wanted to find out that whether I need some money. Those days they used to give $500 only. And he was so affectionate to me. So it's a great pleasure and privilege to introduce Professor Arup. He is the Regents Professor of and Chief of Ophthalmology, University of New Mexico School of Medicine, Albuquerque, USA. His education, as I mentioned, Medical College, Calcutta, India, where he did MBS, and University of Calcutta, MS Ophthalmology. Medical College, Ohio, he went there and did the physiology, PhD in a record time. And Wayne State University, Detroit, MIE, was a postdoctoral fellow, and one of the pioneer of diabetic retinopathy. And he was a resident in ophthalmology in Wayne State University. And Doheny Eye Institute, he was a retina fellow under Professor Stephen Ryan. It was really a prestigious position. He's a Chief Division of Ophthalmology, UNM School of Medicine, Regents Professor of Ophthalmology, UNM, I told you. He was awarded Gold Fellow from FARVO, Association for Research in Vision and Ophthalmology, which is one of the topmost organization in vision research. He received Achievement Award, American Academy of Ophthalmology. He is member of several prestigious societies, including Retina Society and Macula Society. He was the editor of a book, Therapy of Ocular Angiogenesis, Principle and Practice. The photograph is there over there in the slide. He was a member of several prestigious journals, Investigator of Ophthalmology and Visual Sciences Journal, Indian Journal of Ophthalmology, Middle East African Journal of Ophthalmology. His research interest is diabetic retinopathy and novel therapeutic targets. Many of the basic research work, he was trying to find out that whether it can be translated in the clinical research, clinical products. He was funded by the National Institute of Health, Diabetic Retinopathy Foundation, American Diabetes Association, and Veteran Administration of Research and Development. He was the NIH study section, which is a very prestigious one, and JDRF Research Grant Committee. He participated in many clinical trials on diabetic retinopathy, EDIC, READ2, RIDE, DRCR, and Boulevard. You would be knowing more about it. He is a member of Ophthalmology Group, EDI study, Epidemiology of Diabetes, Intervention and Complications. Let's welcome Professor Arup. Thank you.
Thank you, Jyoti, for such a nice, long introduction. It's always a pleasure to be here back to Shankar Nethralaya, and I feel the absence of Dr. Badrinath this time. I'm here after 5 years. I think Dr. Badrinath has been an inspiration to all of you, a dream mentor to all of you, a quintessential hero to all of you, as I have been always indebted. He has been an inspiration in my career also. So with that note, I'd like to start my talk, Beyond VEGF, in search of novel therapeutic targets of diabetic retinopathy. So in this talk, I'm going to talk about the limitations of the current anti-VEGF trials. Second, I'm going to talk about one of the important target molecules, angiopoietin 2, and the story of Vabysmo. And finally, I will talk about some other possible potential target molecules in DME and AMD that is happening in our lab. And finally end with the story of genomics in diabetic retinopathy. And I think the A1C is just a part of the whole story. Maybe there are other factors are present, important. This is my financial disclosure. So again, my tribute, my big salute to the visionary legend, Dr. Badrinath. With my deep regards and homage to him, I start my talk. The story of VEGF starts with Napoleon Ferrara, who was a scientist at Genentech and first isolated and cloned VEGF, an endothelial cell-specific mitogen secreted from pituitary follicular cells. It is an important angiogenic factor. Is present in the primitive animal species. Here is an example of marine jellyfish where the tubular formation is regulated by the same molecule VEGF, just like the blood vessels are modulated by VEGF. And again, VEGF has been targeted by different approaches, starting with Aptamer to anti-VEGF antibodies, Avastin, Lucentis, VEGF trap, siRNA. And we all know that VEGF is a good target molecule. Definitely, here's an example of a good responder of DME patient with one injection of anti-VEGF, you get a great response. This doesn't happen always. I see this, I will say that this is in 1/3 of the patients, good responder. That means after 3 monthly injections, if you look at the CRT, there's a decrease of CRT from the baseline by more than 20%. That's why I define good responder. And that's about 1/3 of the patient. And that has been shown by all the large landmark clinical trials, DRCR Protocol I, same way Vivid, Vista, Read, Rise, all have these trials and shown that anti-VEGF drugs are the first line of treatment of DME. So when Judah Folkman started with this concept of angiogenesis in 1970, he told me that there are, in 1970, if you do a Medline search, there are 3 publications came up in the Medline search on angiogenesis. One was the article written by him, New England Journal of Medicine, on angiogenesis, and the 2 other articles criticizing Judah Folkman, that angiogenesis is not the real story. So that's the way it started. Now in 2024, it's one of the leading agents, anti-VEGF drugs, in the cancer field and in our ophthalmology field. His ideas were scorned by many. When he used to come to give lecture in the hall, people would leave. Colleagues would leave the hall. Students are discouraged from joining his lab. So the moral of the story is be persistent, never give up. So again, not all DME patients respond equally to anti-VEGF drugs. Here is an example on the left, good responder with 1 injection. And here is an example of poor responder with 6 injections. The DME still persists. So this is the 2 extreme ends, and 1/3, and in between there is a group intermediate responder where you see the response, maybe 1 line of vision improvement. So what did we learn from these trials? VEGF inhibitors definitely are beneficial in DME and DR progression, and only 27 to 40%, I would say 1/3, as per DRCRI, RIDE, RISE, Vivid, Vista, shown that only 1/3 patients have an improvement more than 3 lines of vision. Again, monthly injections are treatment burden, and most importantly, what I like to stress today, DME is a heterogeneous disease. It has many molecules involved. It's not like angiogenic disease, PDR, where it is mainly VEGF, or a wet AMD is mainly VEGF. But in DME, it's more than VEGF. So when you look for an ideal therapeutic agent, you look for longer durability so that it needs less frequent injections, and you look for stronger efficacy, and maybe you can look for target other than VEGF. So here is a classical paper from Funatsu, published in Ophthalmology in 2003, where you can see the levels of VEGF is all over. The mean level is higher than the control, But there are high levels of VEGF and there are very low levels of VEGF. So when you are giving a patient anti-VEGF drug in this group, you are not getting a response. Same way, if you are giving a VEGF, anti-VEGF drugs in the higher group, you are just neutralizing only a small part of the VEGF. So its level is variable in patients with DME. There is another interesting paper where it looked at the aqueous humor levels of different cytokines. And if you look from the no DR to moderate to the PDR, as the severity increases, if you look at the VEGF, there is no correlation of the VEGF level with no DR versus PDR. They are not significant. P-value is 0.7. But if you look at other markers like IL-1 beta, 6, 8, MCP1, there is an increasing tendency, you can see here, from the... So many other inflammatory are playing a role in DME. This is an aqueous humor analysis, which may not be truly reflective of the vitreous levels of the cytokines. So DME is more than VEGF. All the features of inflammation, starting with leukostasis, very important step, white blood cells adhering to the endothelial cell, then macrophage and neutrophil infiltration. I'll talk about that a little bit later, and a couple of new targets. And then here is all the cytokines, VEGF, TNF-alpha, IL-6, CCL2, and CXCL1 is another one that I'm going to talk about later. And microglial activation, complement activation, increased blood flow, increased tissue edema. So all the features of inflammation have been described in the animal models as well as human diabetic retinopathy. So these are some of the potential molecular targets that we have been working in our lab. I'm going to tell the story about angiopoietin 2. Earlier, we worked on the proteinases like MMP and urokinase. Recently, we worked on CCL2, and the latest one is CXCL1, interleukin-6 and IL-beta. Kallikrein-kinin system has been done by Ed Finner at Joslin Diabetes Center. So not necessarily all these molecules that we have been working in our lab have been successful when it went to the clinical trials. Only ANG2 is a success story here. And I'm going to talk about that in this part of the story. So when the blood vessels are stable, angiopoietin 1 works on the Tie2 receptor and keeps maintain the stable vasculature. And then there is an angiogenic switch. The Ang2 levels increase, and you can see now the Ang2 now acts on the Tie2 receptor along with the VEGF, which acts on the VEGF-R2 receptor, and both factors work together. Ang2 binding leads to pericyte loss, inflammation, cell membrane instability, and most important one is the increased VEGF sensitivity, whereas VEGF-A promotes leakage and angiogenesis process. So there are three important functions that we need to remember when talking about Vabysmo. Ang2 is a pro-angiogenic factor, it's a vasopermeability factor, and the most important one is it's a pro-inflammatory factor. I will talk about each one now. This is 2003, before Avastin was approved for colorectal cancer. We published this paper on angiopoietin 2. We found the Ang2 level goes up in the retinas in the mouse model of neovascularization. And here you can see, if you see this retina, all these arrows pointing towards the new vessels on the ILM, whereas these animals treated with the angiopoietin 2 inhibitor, and you can see just one maybe blood vessel. So this anti-angiopoietin 2 factor can reduce the retinal neovascularization by almost 86%. That was published in 2003. Then we also looked at its vasopermeability action. You can see on the retinal endothelial cells in culture, if you add Ang2, it induces the separation from the endothelial cells and breakdown of the tight junctions between the endothelial cells. And you can see the electrical resistance between all these endothelial cells goes down with time. So it's an important vasopermeability factor. So that was published in 2011. Then we also looked at angiopoietin-2 is pro-inflammatory. And you can see this is Fini Monicrach in our lab. He did his PhD at MDRF and joined in my lab almost 10 years now. So he showed that with angiopoietin-2, there is, you can see increased endothelial cell monocyte addition, leukostasis. And as well as ICAM1 expression is increased with angiopoietin-2, and then with the treatment of angiopoietin-1 or inhibitor of Ang2, you can decrease this ICAM1 level in the retinas. This is another example where you can see the retinal blood vessels stained with red, and these are the microglial cells stained with the green fluorescent dye. And they are inactive cells with long processes equally distant from each other. If you inject angiopoietin-2 in the vitreous of the animals, this is the picture you see, and this is a higher magnification. There are two things to note, please. One, these microglial cells are now amoeboid, round, less processes. This is activated microglia. So here is inactive microglia, here is active microglia, and all these round cells, red, some of these cells are red stain. These are all monocytes. So there is increased monocyte trafficking. This is exactly happening in diabetic retina also. So there are two things happening. One is the microglia are activated. They secrete different cytokines, including VEGF, CCL2, a lot of, and then these monocytes which become converted to macrophages. So again, you can see Ang2 is pro-inflammatory because it's two things, monocyte trafficking and microglial activation. In patients, you can see, Ang2 level is up in ARMD, vein occlusion, DR patients, PDR patients. And here is a first bispecific antibody designed by Roche Genentech. It's with the two targets. One is the anti-VEGF Fab, one is anti-angiopoietin-2 Fab, and FC is optimized for faster systemic clearance. That is the Vabysmo. And this is the company now has got the drug approved, and I was told that this is now launched in India several months ago now. So the dual inhibition definitely decreases. We talked about already the macrophage infiltration than more than VEGF alone. So definitely it's an anti-inflammatory. The other thing that the company is interested, they are looking at the reduction of fibrosis. Patients with just anti-VEGF alone have more fibrosis epiretinal membranes. I'm not presenting the data here. Compared to those who are treated with faricimab, that's another area they are looking into it, probably through the anti-inflammatory action. So eosinometry Rhine study, phase 3 clinical trials, looked at 3 arms, 6 milligram faricimab, and compared with the 2 milligram aflibercept, there is another personalized treatment interval. So in both these eosinometry and Rhine trial, in phase 3 in DME patients, you can see there is really no big difference between this faricimab, the green one, and the blue one is the Eylea aflibercept. There is no difference in, they are equally, they are non-inferior to vision, in terms of vision gain, non-inferior to aflibercept. However, if you look at the thickness or the fluid, definitely faricimab has more advantages than just anti-VEGF alone. Here is an example between upper panel showing that ARMD patients and the DME patients, definitely with the faricimab, if you look at the Ang2 level, it's not detectable in both groups, whereas definitely with the Eylea, as expected, Ang2 level is still high in these patients. So faricimab definitely reduces the Ang2 level. This is all aqueous levels. And both in Yosemite Rhine and in the DME study and the Tenaya and Lucerne in the ARMD study, definitely there is a more decrease in the central retinal thickness in the faricimab-treated patients. And both studies, Yosemite and Rhine, showed that more than 50% can go without injections even up to 4 months, which is an important finding for faricimab-treated patients. Here is an example of Yosemite Rhine study. You can see almost from 1 year to 2 years, you can see almost 78% patients went up to between the 12 weeks interval to 16-week interval. So you need only every 4 months, that means 3 times a year injection with the faricimab. And this is true for the wet AMD patients with the Tenaya and Lucerne study, same way. From year 1 to year 2, almost 78% patients went up to Q16 weeks interval. And this is now approved for retinal vein occlusion just a few months ago, shown by the Balaton and the Comino studies. And here is an example of one of my patients in the clinical trial. Our center was a part of the Yosemite trial, phase 3. According to the protocol, this is a Boulevard study patient actually, phase 2. Here is an example of a DME patient, and according to the protocol, you have to give every month 6 injections, loading dose for DME patients. So we gave an injection, you can see the decrease in DME. It came back again a little bit. Then once after this, you can see month 3, 4, 5, and the definitely CRT is completely decreased from the baseline. Now, what happens after 6 months? At this point, you are told it's up to the investigator's discretion. And I had this patient, I did not do the injection on the 6-month visit. And it remained the same. 7 months, I did not inject. Vision is still 20/50. CRT is still normal. Month 8, month 10, month 11, we can see some of these cystic appearances and month 12. So this patient went almost up to 6 months without the injection. I wanted to show not everybody responds this way. This is one of the studies in the trial that showed excellent results. So Yosemite Rhine study, the pooled post-hoc analysis. This is at the baseline. You can see the diffuse thickening. And at week 16, from here to here, you can see there is definitely the less thickening, more resolution of the macular leakage. And this is the actual the data showing the measured the macular leakage area assessment, and definitely this blue bar is the aflibercept and faricimab is the green, almost 53% reduction in the macular leakage. Definitely it's a better drying agent compared to anti-VEGF alone. Same way for wet AMD, there is definitely rapid CERAS PED resolution after faricimab. And here is an example of before and after. So definitely less edema, less fluid with the faricimab treatment. And this is the Tenaya Lucerne pooled post-hoc analysis. Again, the purple one is the faricimab, and this is the aflibercept, the bluish bar. And there is a definitely significant reduction in terms of the CERAS PED. Here is an example of one of my patients with the DME. I started with the Avastin. No response, it went up. Second injection, third injection, I did almost 10 injections. And then I switched to faricimab. You can see I started to see the changes in the first injection. That doesn't happen always. You need at least 2 to 3 injections to get the results. And here is the faricimab second treatment, you can see definitely. So some of the patients, I would say 40 to 50% of the patients who do not respond to the anti-VEGF drugs, I see the response with this treatment. Here is an example of a CRVO patient where you can see we have been trying Eylea, several injections. I was actually waiting for the Vabysmo to be approved because in the USA, Vabysmo was approved later on. So with the one injection, you can see you did not see much response, but the second injection, you can see after 2 injections, we see the vision improvement 20/20 with the second injection of Vabysmo. So definitely there is a concern about retinal vasculitis and vaso-occlusion. As you compared, sorry. You can see the vasculitis, vascular occlusion is more in the brolucizumab group, but we are seeing the same thing in other drugs, aflibercept and ranibizumab, but much higher in the brolucizumab. And USA, at least, very few retina physicians are using brolucizumab these days now. So again, with faricimab, we are seeing there is a decrease in the Ang2 levels, is an effective disease control and clinical durability up to even 16 weeks to 20 weeks. And here is the story that when we started the work, we did not get any support on the Ang2 work. We published our work in 2003 and 11. Our NIH grant was rejected in 2012. And then in 2016, 3 drug companies, Genentech, Regeneron, and RPO started clinical trials. It is now approved in 2022. So the lesson is never give up. Keep trying. So I'm going to briefly talk about 2 other molecules that are important target molecules before I go to genetics. One is the increased expression of, you can see in the CCL2, which is the chemokine ligand 2, is very high in the retinas, in the diabetic retinas. And what is CCL2? It's a molecule also known as MCP, monocyte chemoattractant protein. It attracts monocytes in the tissue, and I showed earlier that when you inject Ang2, there is increased monocyte trafficking. Same thing happens in diabetes. And it's an important pathway for monocyte infiltration in atherosclerosis, neuroinflammatory disease, Alzheimer's. And here is an example of diabetic retina. Earlier I showed Ang2 injection. Here is an example on the right, on this left here, microglia. No monocytes here, but here are all the monocytes you can see around cells, infiltration, and the activated microglia. So if you target CCL2, which is MCP1, you can use knockout animals, and you can see almost significant decrease in the monocyte infiltration, and the microglia are actually not activated. So it's an important target for diabetic macular edema maybe. So we used an inhibitor against the receptor for CCL2, which is the CCR2 and CCR5 inhibitor. You can see the vascular leakage as shown in the green bar with the drug treatment. There's a decrease in the permeability and decrease in the junctional molecule in diabetes, and this is recovered with the drug. So this is an effective target. However, a clinical trial was started with the Pfizer group and did not find a significant difference in the human DME patients. And I will tell you the story later why. Second molecule, so we talked about monocytes. Now we are interested in neutrophils. And we think that neutrophils play an important role. Although these neutrophils are known for acute inflammatory disease, they're important for diabetic retinopathy pathogenesis. And our discovery was incidental. We found out that these nets, the neutrophils, release these molecules called nets, neutrophil extracellular traps, which is basically a network of DNA and granular proteins that are important for cell damage, infection, sepsis, diabetes, inflammation, atherosclerosis, ischemia, is very important thrombosis, vaso-occlusion, and also fibrosis in probably in the epiretinal membrane formation. So our goal is to look at this role of neutrophils in diabetic macular edema. why we got interested in this molecule. We found out if you use retinol endothelial cells treated with the high glucose, there is significant increase in the expression of genes like CCL2, which we have shown before. But also we found CXCL1 is a chemokine molecule. And that leads to this study where we found out what is CXCL1. It's a chemokine that attracts neutrophils. So why neutrophils are so important? So definitely the CXCL1 level goes up and the neutrophils come in the diabetic retina, you can see. And if you inhibit the neutrophils with this antibody, Lys6G antibody, you can decrease in infiltration of the neutrophils. So neutrophils as well as monocytes infiltrate in the retinas in the diabetic animals. And you can look at the neutrophil depletion. If you can deplete the neutrophils, it also causes significant decrease in the protein expression of these two different markers, netosis markers, H3 and PAT4. So our goal is to target CXCL1 chemokine or maybe that neutrophil extracellular traps so that we can prevent inflammatory changes. So this is a new concept we are working in our lab. And this is our hypothesis. The neutrophils release the nets that causes the increased vasopermeability. As you can see, with the nets, there is a decreased VE carrier, that means there is increased permeability. And we have shown in this study that with high glucose treatment, more and more the cells become, neutrophils become netosis process undergo with the high glucose treatment. So again, neutrophils are important cell mediators. This is present in a human diabetic retinopathy patient. These are stained with these are the neutrophils in the tissues you can see compared to the non-diabetic retina. So again, this may be an important target if we target CXCL1. So what are the targets molecule in the pipeline? We talked about, this is just approved, high-dose aflibercept. We know about, this is a clinical trial going on, suprachoroidal injection of triamcinolone. This is a molecule I'm going to talk about later, VEGF C and D. These are important mediators for permeability and angiogenesis. OPT302, there is a clinical trial going on targeting VEGF C and D along with VEGF A with Eylea. This is another molecule, KSI 501. They're targeting VEGF and IL-6 together. Tyrosine kinase inhibitor will be an important story, and anti-integrin and inhibitor of kallikrein. These are some of the clinical trials going on and would be the potential targets. So the next few minutes, I'm going to talk about the genomics of retinopathy. As I showed to you, some of the drugs that we tried against MMPs, against CCL2, did not translate into the human diabetic retinopathy patients. Why? Maybe the dose used in the trials may not be the optimal dose humans. Though for systemic drug, maybe it did not reach the retina in enough concentration. Or the most important reason may be the pathway in the animal models may not be the same important for pathogenesis of DR in the human. So whatever you are thinking that this is showing in the animal results may not be the same mechanism in the human. So there could be the genetic variants that may be important, and maybe we should do this way. Instead of going from cell culture to the animal models to the human diabetic clinical trials, maybe we should learn from the human patients and go backwards to them, find out the exact variant, and go to the animal model and cell culture model and come up with the novel therapeutic targets. And I will show you our preliminary findings on genomics of diabetic retinopathy. So as you know, the classic ETDRS study showed that no DR goes to mild NPDR, to the moderate NPDR, to the severe NPDR, and then PDR. But not everybody goes the same way. Some of the patients, and the macular edema can occur in any stage of diabetic retinopathy, wherever in the moderate to severe or PDR, not necessarily in this order. It can happen independent of this stage, this course. And also a small number of patients do not progress further. We see always, and you see always, the patients where there are 30, 40 years of diabetes, and there is no retinopathy in the eye. In spite of A1C, not necessarily the good control. A1C is high. So this is we call extreme phenotype, whereas we see only a few microorganisms or no retinopathy at all. And I'll talk about this group now. So the question of diabetic retinopathy, another question that we asked in the genomics is that are PDR and DME two different disease processes? And why we say that? Here is an example of a patient with a big NVD, but there is no DME at the same time. So this patient, in spite of high VEGF level in the vitreous, is free of macular edema. So that's the surprise why the same VEGF level is not causing macular edema. Same way, if you look at a DME patient, there is an increased VEGF level, there is no NVD or NVE. And this is an example of a patient with PDR, day 1 post-op. You can see all the laser marks, but the macula is completely free of edema. So we did a retrospective study looking at the coexistence of the PDR and DME. You can see in PDR patients, the concurrent DME was only present in 15%. 85% of the patient did not have the concurrent DME. Same way, in DME patient, only 20% patients had angiogenesis. Almost 80% patients did not have any angiogenesis in spite of the high VEGF level. So these tells us there may be different genetic factors that make one susceptible to PDR and one susceptible to DME. And we are actually in the middle of finding some of the variants in these two phenotypes. So question remains, does ethnicity play a role in the phenotypic heterogeneity of DME? And we found out very interesting. This is a patient in American Indians that we have in our state. These American Indians, they develop just a few hard exudates. Majority of the patients like this, very non-center involving DME. They rarely need anti-VEGF injections. So they are getting focal type, non-center involving DME, where you don't use anti-VEGF injections. Whereas in the Hispanic group of patients, where you see extensive macular edema, center involving macular edema, and they need the anti-VEGF injection. So why there are these two types of DME, you can see focal versus diffuse, present in two different important ethnic groups, predominantly in native Indians, predominantly in Hispanic. So there must be some kind of genetic factor who gets extensive DME, who gets mild DME. Here is an example of a diabetic patient, 51-year-old type 2, 35 years of diabetes. A1C is not that well controlled, 8.5. There's only one microorganism here. So why this patient is protected from diabetic retinopathy? So not everybody goes blind. This is the extreme phenotype. And we see this kind of patients and we looked at the genomic sequence. So the other question is that in Wisconsin epidemiological study, Ron Klein showed that retinopathy PDR, by 15 years, 100% develop any retinopathy, but only 50% develops PDR. Other 50% will never develop PDR with a longer duration of diabetes. So question arises, are some diabetics are more susceptible to PDR angiogenic process, and these 50% do not develop PDR even with the longer duration? Is there a genetic factor present for that? And this is for the type 2. So my question is that I know that A1C plays an important role, and that has been shown by the landmark studies, but it doesn't explain always the phenotype of diabetic retinopathy. Here is John Latchen's paper of DCCT trial post-hoc analysis, where he concluded that 11%, the total glycemic exposure, A1C and duration, explain only 11% of the risk of developing DR, according to this calculation, which goes over my head. Is a very complex statistical analysis. He's a statistician, and he showed that from the DCCT study, this glycemic exposure explains only 11%. And in that paper, they talked about other factors, genetic, presumably explain the remaining 89% of the variation in risk. So again, I'm not against A1C. A1C is very important, but A1C has to be also have a genetic factor modifier to have the severe disease. So this is a story from Jocelyn Diabetes Center, where they have 50-year type 1 diabetic patients. You can see, again, 50% patients develop PDR, other 50% never develop, and about 40% of them had no DR or myelin PDR in that cohort. And they found out hemoglobin A1C unrelated to complications. Very important, interesting finding. And they have related this finding to this factor, retinol binding protein 3, which is increased in the retinas of the patients who are resistant to diabetic retinopathy. It came out in Science Translational Medicine. So in our Divergent study, we asked 3 questions: Hispanics and urban American Indians in Albuquerque with Caucasians at Jocelyn Diabetes Center. We looked at why are some people protected from DR in spite of long duration, why are there genetic variants for PDR and DME, and finally, are the genetic factors for anti-VEGF response good versus poor responder. I will just share just some of the preliminary findings. Again, as you know, that only 1% of the Northern Europeans carry a mutation of the CCR5 gene, which is the CCL2 receptor, 5 receptor. And those patients, if you have this mutation, you are lucky. You will never get HIV infection. You are protected. So question is the same way. If you have a mutation in this gene, PCSK9 gene, you can have a lower LDL level and protected from heart disease. So do you have the same kind of genotypic finding in diabetic retinopathy? So we looked at the one end extreme phenotype, other end the severe PDR, advanced phenotype. We are not talking about small NVE. These are all NVD patients. Here is a one cohort extreme phenotype, about 103 patients, 38-year diabetes, not a single microaneurysms in either eye. Whereas on the other extreme, we had about 176 patients. This is between us at UNM versus and Jocelyn Diabetes Center, where you can see these are advanced PDR. Again, we did not include any NVE patients here. So this is our method. These are all type 1 or type 2 diabetics, duration more than 20 years, no diabetic retinopathy or 5 macroaneurysms or less, and in the other extreme, the PDR active NVD. And we did the whole genome sequencing. And if you look at these 2 cohorts, you can see there is no correlation of the A1C. If you look at this no DR group, you can see the less than 7, that is the normal level, was only present in this blue bar. The majority of the other patients are having A1C higher than 7. In the same way, in PDR patients, where you expect them to be A1C more than 9, was present in only 1/3 of the patients. And this other group, you can see even 20% have a A1C level less than 7. How do you explain that? So this is our case control study, whole exome sequencing, exome-wide association analysis, then adjusted for age, sex, ancestry. And we also did rare variant analysis, and we then did some functional validation. So here is the finding of our Divergent study. In the no DR cohort, we looked at about 8,400 variants in 103 cases of no DR, compared with the 176 PDR patients. And we found only one single variant in this gene called EHMT2. And I'll tell you what is it. That shows significantly higher. It's present on chromosome 6, and it's a loss of function variant. So what is the AHMT2 gene? It's a euchromatin histone methyltransferase 2 is important for development, cell growth, metastasis, but is an important factor for angiogenesis and cell migration and proliferation. So this is an angiogenic factor, and it is present expressed in the rod photoreceptor, amacrine cells, Muller cells. So question is, it is located on chromosome 6, which is an important area HLA locus, which has about 60 genes. This is the HLA-B. Here is the TNF-alpha gene. Here is the heat shock protein. Here is the complement factor 2 and 4. And here is our EHMT2 gene is sitting right there between the TNF-alpha, next to the TNF-alpha and HSP. Whether it is close proximity to the TNF-alpha and complement 2 plays any role, we do not know. But definitely we know this is an angiogenic gene, very close to the TNF-alpha on chromosome 6. And our hypothesis is that in diabetes, EHMT2 causes increased expression of genes, angiogenic gene response, angiogenesis, PDR, whereas if you have a mutation of EHMT2 gene, you have a decreased expression of the genes on angiogenesis and inflammatory response, maintain cellular homeostasis and protected from DR. Whether it is going to be true, we do not know. We have a lot of studies on function and validation. The second analysis we found there are several rare variants were enriched in the no-DR group. These are the 5 top genes we found out. Toll-like receptor 5, ribonuclease, L, stromal interaction molecule, EDA2R, OSBPL1. What are these genes mean? These are all important for immune signaling and related to TNF-alpha and IL-6. You can see these 5 genes that we found out. Just remember that these are all related to with the network analysis related to the TNF. Again, we showed EHMT2 is close to TNF-alpha. The story is coming after TNF-alpha and IL-6 again. So again, all these variants are potential loss of function, probably modify the phenotypic outcome. And when you do the ELISA study in these patients, shown by the blue bar, the no-DR patients, you can see compared to the PDR, there is a lower VEGF level, lower VEGF-C level, lower angiopoietin-2 level, lower CCL2, lower TNF-alpha. So all the inflammatory mediators are lower in this protected extreme phenotype group. So it's a less inflammatory phenotype. So conclusion from the study that modified genes may determine the susceptibility or protection. EHMT2 gene is significant in the no-DR, and the rare coding variants related to the TNF-alpha and IL-6 in the no-DR. There's a loss of function, less inflammation, and protection from diabetic retinopathy. So I want to end with these few slides on the good responder versus poor responders. We have a lot of work to do. Maybe we can collaborate with Shankar Nethralaya on this study. This is a very small number of sample size we found out. There are several genes variants in the poor responder. And one of the things I want to stress that VEGF-C is an important mediator. And there is a clinical trial going on targeting VEGF-C. So not getting into the too much details, I'm going to showing here the good responders versus poor responder. Good is the blue, poor responders is, you can see all the poor responders shown by the orange bars here have a higher levels of VEGF-A, VEGF-165, VEGF-C, VEGF-R1, angiopoietin-2, CCL2 rate. So that means this is a more inflammatory phenotype in the poor VEGF responders. So my question is, are these cytokine levels are up in this group of patients? Here is an example of a case 1. You can see there's a poor responder with the anti-VEGF injection. There is no response. When you did the whole exome sequencing, we found a variant in the VEGF-R3, which is the receptor for the VEGF-C, and we measured ELISA level, high level of VEGF-C. Again, apart from the VEGF-A and B, C and D are now shown to be important mediators of vasopermeability factor in DME. Here another example of a case 2 where, where you are seeing a poor responder with anti-VEGF injection. When you look at the whole exome sequencing, there is a HLA-DRB variant and there is increased level of CCL2. So why don't the DME patients all respond to the anti-VEGF drug? This is my explanation. Poor responders could be due to the, again, as I showed, high VEGF level. So if you are injecting an anti-VEGF level, you are not neutralizing enough VEGF, so that's why there is no response. And there may be also VEGF-C level is high in this patient, and you are not neutralizing VEGF-C. In fact, anti-VEGF-A drugs causes a compensatory increase in the VEGF-C and D. Other one could be a possibility that there is inflammatory phenotype, there is increased level of CCL2, Ang2, RAGE. So you are not neutralizing those factors. And remember, anti-VEGF agents do not cause reperfusion. So hypoxia is present still there, VEGF secretion continues because there is no reperfusion. And there is a compensatory VEGF increase after each injection. So here is an example of this VEGF-C/D inhibition. Here is an example of a molecule with Eylea targeting VEGF-A, VEGF-B, and placental growth factor. Lucentis acts on VEGF-A only, whereas OPT-302 is a VEGF-C and D inhibitor. So when you are combining a drug with Eylea and OPT-302, you are neutralizing all the VEGF forms. So this is a pan-VEGF inhibition, and you can see VEGF inhibition itself upregulate VEGF-C and D. So there is a clinical trial going on. It's called sozinibercept, and just same way aflibercept, we call it. It's an Indian name, looks like to me. Sozinibercept, a VEGF trap molecule. But problem with this is that there are two injections needed, one anti-VEGF-CD and one aflibercept, which is not convenient. But this is a clinical trial, has shown very good results in the phase 2 in the refractory DME. I think there was 3 lines of improvement versus aflibercept, 2 lines of improvement. And they are getting into the phase 3 trial, SHORE and COAST trial, looking at the combining ranibizumab and combining aflibercept. So in future, it will be a contrast to one-size-fits-all approach. Maybe one patient has a high VEGF level, and the other patient maybe have a low VEGF level here. Maybe Ang2 is higher in this patient. So it's a mixture of, each patient is different from each other in terms of the molecular signature, and maybe a different, that's why we are seeing different differential responses to anti-VEGF drugs. So again, the last 16 years of anti-VEGF therapy, many questions are unanswered. That leads to the agenda for how anti-VEGF may be enhanced to improve patient outcome by solving the drug resistance, by solving the novel biomarker for anti-VEGF response, combined therapy targeting beyond VEGF. And with that note, I'd like to acknowledge my collaborators in the Regents study at UNM, Jocelyn Lloyd Ayalo, Jennifer Sun, the whole TGen group, Shaurya Anand at NEI, Mark Horton. And these are my students and fellows over the years and my funding sources. Thank you.